Could you? Awesome. So hello, everyone. Welcome to our last talk in the Women in Network Science seminar for the fall term, so also the last talk until the end of the year. And we decided to end this seminar series for the year with a bang. We have as a final speaker for this year, Professor Heather Harrington from the Mathematical Institute at the University of Oxford. She's also a Royal Society University Research Fellow, and she leads the Algebraic Systems Biology group. She's got quite an impressive resume of accomplishments, and I'm just going to mention a few here, which are that she's been recently, as of 2019, been the winner of the Adams Prize of the University of Cambridge, or a co-winner of that on the topic of mathematics of networks. And she's been awarded the Philip Leverhulme Prize in 2020 for advances on analysis of noisy data. And I'm very, very excited to hear from her about the new work that she's been doing in the space of topological data analysis on higher order systems. So Heather, please take the stage. It's yours.
Great. Thank you very much. Thanks for the invitation. It's really lovely to speak with you. I think the Women in Network Science, that this is a great community, and I'm really happy to join it. So today I want to talk about some work that was actually led by female mathematicians. So on my title slide, the preprint is called Hypergraphs from Multiscale Cycles in Structured Data. And you can see Agnese, who's all the way on the right in this picture. So this was taken in an Oxford college. So Agnese is all the way on the right. And so she was a Hooke Fellow here in the Mathematical Institute, previously my student, Ph.D. student, and she's now working with Michael Stumpf in Melbourne. And Iris Yoon, who's also, she's in the middle of the picture here. She is a postdoc in the Mathematical Institute, and I'm one of her mentors. So Christian, who is the middle author, here you can see he is a PhD student in Melbourne with Michael, and Deborah, who is part of our Algebra in Africa initiative. So we had a grant that supported activities between African mathematicians. And so during one of these activities, she became interested in some of these things. So it's really a multi-disciplinary collaborations. I'm really excited to talk with you about it today. So we've developed a pipeline and we've called it Hyper TDA. So before I get into all the details, I want to start with advertisements that we have 3 one-year postdocs. So the end of our Center for Topological Data Analysis is sadly approaching, which means we have funding that are all fixed one-year positions. And we would love for anyone that's working in TDA to apply. We have 2 open, kind of open call on applied maths Hooke Research Fellowships. I came to Oxford on a Hooke Research Fellowship. It's great. Please apply. I'm not involved in that interview, so I can't give you much more information. I'm not on that panel, but it is across all of applied mathematics. And finally, we have a Women in Computational Topology workshop. It's the 3rd workshop, and we're holding it in Switzerland. And for those women in network science, I would just say that these types of events are great. If you're also a computational topologist, I would encourage you to apply. The deadline is the 1st of December. Okay, so that is the advertisements while everyone's very focused on the talk and the beginning of it. But I want to have a couple motivation slides, which is the type of data that we're looking at is long entangled open curves. And these spatial curves are piecewise linear, and some examples of them come from biology. And for example, you could have knotted curves. These are open knotted curves in space. And we want to study the geometry and the topology. And there are many methods from low-dimensional topology or from some standard approaches to study, for example, knotted proteins, which is something that we looked at with students in a previous project. But there's many different methods looking at the radius of gyration and curvature and average Gaussian number, looking at ways to study, for example, knotted proteins to close them or to project them with these knotoids. So there's lots of different ways to study these open entangled curves. And they do give you a lot of information, but one of the cons, the negatives there, is that these can be require really precise data. And why do we care about these long entangled biopolymers? Is that they're really important in the shape of this data has a very special, is thought to, the shape of the data affects the function, but understanding that relationship is really complicated. So for example, folded chromatin, so these are really long DNA molecules, and the data we have for those are very noisy. We're looking at proteins, we have the PDV, the protein data bank, and these can fold complex shapes. And it's, for example, known that knotted proteins, they don't degrade as easily, right? So there's this question about what is the role of, for example, these open knotted curves that are proteins, and as kind of a motivation to start how we got to hyper TDA. I first want to kind of briefly talk about some work that we looked at with 4 undergraduate students. And so that was looking at, oh, whoops, let me see if I can go back. Yeah, so that was looking at, if you take there's a database where we know that a very small fraction of proteins are knotted, and we had a very simple question, which is that, can we distinguish knotted proteins from unknotted proteins with topological data analysis? So this is where we can think of this piecewise linear curve. We can look at each of the carbon atoms as the vertices of our curve. And so maybe as a network scientist, we think, well, this is a very boring network, right? It is simply, it's just a curve in 3 space. But if we're looking at the vertices here, we might want to ask something about the geometry. And so one way to do that is to think about this problem and study it with topological data analysis. And so this is an undergraduate project, and we said, okay, let's take this data and we build a higher order network. We build simplicial complexes on the data simply by taking each data point, and you thicken each data point. And then you look at the whether these 2 thickened points are within a pairwise distance. You can think of it like pairwise distance. And if they are, then we would draw a 1 simplex between. So you think of all the vertices as 0 simplices, and we can draw 1 simplices. These are edges. And then if we have 3 points that are all within pairwise distance, we would draw a triangle. And so you can build a filtered simplicial complex. So we build this sequence of spaces on this data. So and then we compute homology. And what we're looking at in the protein setting is computing persistent homology. So because we can look at the sequence of spaces and compute homology. So these are the number of connected components, the number of one-dimensional loops. In our case, we're looking at one-dimensional loops. And we can then see when do we have, at what value of t, so you can think of what distance do we start to have these topological features appear. So, and then where do they then get filled in? So you can see kind of from the middle, value of t to the one on the right, everything seems to get filled in. So we would have no loops, no one-dimensional features here. And the output of a persistent homology algorithm is a barcode or a persistence diagram. And so a persistence diagram on the right is going to show we have points, x and y coordinates, and the x coordinate says at what point, what value of t is this topological feature, so this loop born in this data, and at what point, what value of t does this feature disappear. And so we can look at the diagonal would be 0, 0. And we can see that, the diagonal is basically, we can make this, we can look at all the points off the diagonal to see the persistence. So we can see the value x at which something appears, and the value y at which, for example, a loop here disappears. And so any points that are very far from the diagonal are very persistent. That's a larger loop. Okay, so that's my introduction to persistent homology, that we get this output of a barcode that gives a summary of all the topological features across different scales. So in some way, it's a multi-scale summary of here one-dimensional features. Now, I see now a lot of comments in the chat, but I don't know if that's because people have questions, or I've just now had the chat come down. Are there any questions?
I think these are all from previously people have been responding from where they're watching.
Okay, great. I think when I went back and forth very quickly, the thing came down, so I can't see the chat, but I can see there are comments. Okay.
So I'll have an eye on it and let you know if there's questions in the chat.
Thanks. Okay, so there were no questions. I'm happy to clarify anything as we go. Yeah. Okay. Great. That is persistent homology. So a maybe nicer picture is we have the input is a point cloud of data. As many of you know, we could also take networks and filter networks. So instead of thinking of building this simplicial sequence of simplicial complexes, we could also start with network data. In this case, we're just starting with, I don't want to say vanilla, but the standard persistent homology pipeline. We use Vittorius-Rips sequence, again, looking at the pairwise, if 2 points are within some distance, if 3 points are all within some distance, then we draw a triangle, etc. And then what do we get out? We get out this persistence diagram. Okay, great. Now, from persistence diagrams, there's different metrics. There's different ways we can look at distances. And there's also ways that we can vectorize it. And so there are a number of different vectorizations so that we can then, for example, take an average. If we have many proteins or many piecewise linear curves, we want to take an average, or we want to, compute something. How are they different? And so we can do that with certain metrics, but we can also then vectorize them. For example, there's persistence images. Here today, we're going to use persistence landscapes. And the idea with persistence landscapes is that we would build from the diagonal. If you're looking at the persistence diagram from the diagonal, you can build basically little tent functions, and you're mapping the persistence diagram, these multi-set of intervals, into a Hilbert space, so then we can take averages. And so here on the left, you can see the top left is the input. This is, for example, our knotted protein. And then we compute persistent homology, and we get a persistence diagram that's in the top right. And then we can use these topological statistics, the vectorization method. And here we've used landscapes. And the idea is now the landscape for a diagram is equivalent. So this is an equivalent representation. But what's really powerful is then we can take averages. So let's suppose I take a class of trefoil, which is the simplest non-trivial knot, and we take a class of trefoil proteins and want to compute an average. And we can do that for a class. They're called homologous proteins. They are sequenced very similar, but we know that some of these are knotted and some of these are unknotted. And so that's exactly what we did. Well, this is what 4 undergraduates did. And you can see on the right, they computed persistent homology. They then mapped it into a nice vectorization where they have knotted and unknotted proteins, and they took an average. And there's different landscapes, which are basically looking at kind of the most persistent, and then the next persistent, and then the next persistent. So the longest bars and the next longest and the next longest. So the second landscape, as we can see highlighted, the second landscape for knotted have a little blip here, right? And so we looked at the averages, and we can see there's this difference. So then the next question we had was, well, can we go back to the data and see where is it different? Now, this is something that's very exciting and relatively new. So most of the time when you have some point in your persistence diagram, it's not easy to map back to the original data. And computing the cycle representatives or the homology generators in an efficient and kind of automatic way has only been possible recently. And that's thanks to what are, for example, different software. One of them is called Irene. And Irene is once it's found a point in your persistence diagram, you can say, okay, what point does that correspond to? And in the original data, and then it gives you all of the vertices, all the nodes back in your data set. And so we can compute the generators. And so that's what we looked at. So in the middle here, you can see that there's a purple generator and a red generator. And what Inez had done was she put the two proteins that are they're homologous, they're almost exactly sequence similar homologous. They put them overlapping so we can see the exact difference is what is in the light pink. And that's what basically the crossing change. That's the difference between where one class of proteins are knotted and one class are unknotted. So we can actually localize the difference in the knottedness, all because we went back into this data and looked at the generators. Now, computing generators is not unique. And so, and I'm going to show you that in just a minute, or at least give you an example. But so far, everything I've told you was already known with other techniques, right? So, but what isn't possible is if we add some noise. And then we asked, if we add noise, can we still get the difference back in finding this knot versus unknot? And so we looked at a number of things, and this slide maybe has too many things on it to be able to explain. But the point is that more or less, the persistent homology was robust even when they added a decent amount of noise to the curve by moving all the points, for example. And that's really what's motivated us to develop this hyper TDA pipeline, was saying, okay, we can take our spatial curve, right, these ordered points in 3D, and compute a persistence diagram. And then instead of just taking one point in our diagram and then saying, where does this map back in the original data? Where is that topological feature? Which points are part of that? Why don't we look at all of them? And that's what we said. Okay, so we can see the most persistent feature, for example, is that big cycle in pink, right? But then the next persistent feature is looking at the purple and the green. Those are the next persistent features. And we can do that. So what you get for each generator, each point in your diagram, you get a generator, and a generator is a set of vertices. So that's just to give you an example. So, just repeat the process. You start with a point cloud. You choose a filtration. Here we use the standard Vittori strips filtration. Those of you who have seen in network science using, for example, a weight rank clique filtration on a weighted network, or a weight filtration on a weighted network, or other types, choose whatever filtration F you'd like. You will get some persistent homology, a diagram. And then you might say, okay, well, where does this point correspond to? And those are your generators, and you can do that for each of them. Okay, so there's some issues with generators. One of them is that, which is kind of motivate on the bottom. It's great if you have different features, different cycles, and you can pick the vertices out that describe them, which is the top. But what happens, which happens a lot, is that you have a hole, a one-dimensional hole in your data. Well, you could probably pick many different possible paths, points that would take you through and circle that. And so generators are not unique. We used Irene, and this is a slide thanks to Greg Hanselman Petrusik, and he gave me a little bit of information I'm going to tell you about Irene, which is that the generators are not canonical, they're not optimal, which is that they're not the tightest or minimal, smallest. And the problem is that computing generators is an NP-hard problem. But there's been some work on, for example, minimal cycle generators. And so Iris, who spent more than a month coding up this jump minimized generator, this is pulling out these vertices, part of a feature, which minimizes any jumps in the data. So you can see in the third panel that the pink vertices don't pick up all of the consecutive points in the data, right? And so there was a work by Lee et al. It's actually a very substantial mathematical work. And they computed, it's a bit technical, but some of the computations with this integer programming problem, it's a NP-hard, but they're able to, with some conditions with rational coefficients, be able to compute minimal generators, and most of them will still satisfy certain properties so that they can use this for minimally generating the points. And then Iris built on top of that this jump minimized generator. So you could get all the vertices in this line set included. So then our next question was, well, we know generators in general are not unique, and these are often not analyzed in data sets. How does it look if we run our minimal generators on the data and our matroid generator and Irene's matroid generators? And we looked at it and we thought, well, actually, these points look pretty good by eye. But of course, we want to do something more to study this. So well, I guess we could have said, okay, well, you could have looked at how many points are overlapping or intersecting which vertices with different possible generator methods. Instead, we thought, okay, hold on, let's just step back. And how would we analyze this data? Because what we're finding here is we're finding points that are persistent. So the cycles are persistent. But we're also finding ones that are less persistent, which are off the diagonal. So what we decided is we take all of these points and map each generator to a hyperedge and create a hypergraph. So the idea is that if you look at the persistent homology, the persistence diagram, you have some choice of a generator. So you get your vertices in your original data. Then the edges are the edges for each hyperedge in our hypergraph corresponds to one of the generators, which is a list of vertices. So a hyperedge is just a set of subsets of your vertices, right? So we don't have the restriction which we have with simplicial complexes. You don't have to include all the lower dimensional faces. We just have sets. So our hyperedges are sets with, they could be different numbers, and they are different numbers of vertices. So it's not uniform, it doesn't have any of those nice properties. And then we look at a weighted hypergraph. So if we didn't have that initially, but we added the weighted hypergraph. And this is a map from your edge to a real number. So that is your persistence. So you have how persistent is that hyperedge. So for example, we see the green point in our persistence diagram is the most persistent. So that has the largest weight. And then the next largest is the red one, and then the one closest to the diagonal, as you can see. And that is just simply the difference. We can take the difference between, y minus x, basically. So we have x comma y for our persistence diagram. We're just doing y minus x. That gives you your weight. Okay. So we have a weighted hypergraph now, and that encodes all the higher order information in the data. The higher order, the homology, the persistent homology in this data structure.
Yeah, just a quick clarifying question. So
Yeah.
you're transforming, using the generators, you're transforming your barcode diagram into a weighted hypergraph. And so you have
Yes.
the same number of points in your barcode diagram as you have hyper edges in the hypergraph.
That's correct.
Okay, thanks.
Yes. But we have more information, right? So normally, in some way, a persistence diagram, while the algebra underneath it in the persistence module encodes a lot of it, we forget it, we visualize, and we just only remember the birth and the death, the X and the Y, and we plot it, right? But we know that X and Y, right, that really is telling us something. We want to know what's happening. We want to interpret what's happening. The way to interpret it is to go back to the point cloud, right? And so we are encoding all of this, higher order information. So just because what you see, what points are necessarily close in your, for example, if you just did clustering on the data, that's not what we are putting in the hypergraph, right? So we're encoding more information in the persistent homology hypergraph. It's really what are the set of points that give rise to a topological feature. And that's what we call the pH hypergraph. Yeah. Are there any other questions? I know this is a bit it's a little bit to get your head around. If there's any questions, I'm happy to answer it.
I don't see any right now, but we can come back to that later.
Great. Okay, so we have our pH hypergraph. This is how we are going from the standard persistent homology pipeline and extending it. And this is cool, right? I think there's a lot of opportunity, and that's partly why I was very excited to talk about this, because now we can analyze, right? So I said, well, okay, well, we can generate different types of, we can choose different generators. But then how do we make sense of them? Well, we need ways to measure them. And what we have in network science is lots of network measures. So 2 of them that we propose, and we tried a few, which I can't remember all of them because, it's like off the page on and on in notebooks. But 2 that we tried was pH centrality, so the importance, which nodes are most important in the original data, in the topological features. So which nodes in my data occur in most of the generators. So those are the key nodes for the higher order information, right? And that's the centrality, that's importance. And then pH communities. And we started this work, and it seems like in the last year, more and more ways to compute community detection of hypergraphs has come out. So I'm sure there are more sophisticated ways than what we've done, but these are 2 things that we're looking at. Okay, so now what do? I say, okay, this is our hyper TDA pipeline. So you can pick a different app, you can pick a different filtration, you can pick a different choice of generators, you can pick a different choice of analysis. With what we've picked, we then look at the minimal generators, we look at the matroid generators, and then we can compute a correlation between our centrality vectors of these 2. And we find actually it's not too bad. There's about 70% as we change the length of different curves. So we looked at 800 random curves. And then we also computed the mutual information between the community partitions that we have with each of these generators. And they correlate, they're highly correlated, which is good. If we had no correlation, well, you'd think, okay, we're done. There's no hope. Each approach will give us very different things, or we'd have to use the minimal generators. the computing minimal generators is more expensive. It takes more time than the matroid generators. So it's easier to use a very fast algorithm where there's theoretical guarantees with the persistent homology using matroid theory. And we used that. So then we could then do all of our analysis with the matroid generators. For completeness, we actually redid all of the results I'm going to show you with the minimal, but they give very similar results. That was just because I was wanting to be very comprehensive here, but actually you can see they do correlate. Okay, so then I'm talking to people that are interested in networks. And so you might ask, well, tell me which centrality did you use? Or at least that's what I would be asking, right? So we used eigenvector centrality for hypergraphs. So it's a paper that came out last year, right? So this is very recent. This was we ranked the nodes in our hypergraph according to the hyperedge membership. So which ones are kind of connected, with the importance of the connections and the weight, right? So if you have nodes that are involved in very persistent generators, they will have a higher weight, a higher centrality. So it does look at both of these. And as many of you know, this is a spectral centrality measure. So we're looking at, and so, oh yeah, in my picture here, yellow, for example, is the highest centrality. And for example, purple has the lowest centrality value. So we get this ranking. So that's the node centrality that we use. And that's the paper that we based it off. And then to give you an example, these are two hand-drawn curves, just to give you an idea of what pH centrality will pull out. So on the left, I have a hand-drawn curve of a flower. And you can see the high centrality values are the ones with yellow. And then similarly, I've got a roller coaster, and you can see the ones where you can see the loop, what looks like they're the loop, and another one that looks like a loop. Those have high centrality. Okay. And then we said, okay, well, let's apply this on real data. So we looked at worms, at nematodes. And this is work that Michael Stumpf had published already. So this was data already available. And we ran pH centrality. So we ran the hyper-TDA pipeline and looked at centrality. And what did we find? If we, each of the nodes, right, we color them, rank them according to their centrality. And the looping behavior had the highest centrality. Very clearly, you can see. And this is how they all look. And then the worm goes through a different behavior, which is called relocation, that has low centrality. Because as you can imagine, there aren't loops that are involved in many different topological features. So that has low centrality. So we can kind of understand it. We can interpret what's going on because we can go back to the data and see it. And then the intensity of local search, which has looping, but maybe not as persistent because it's smaller looping. So it's not as high centrality, for example, as the major looping behavior. So that is one example of a worm moving. And we use this hyper-TDA to really interpret what we are observing in that data. Okay. Then we look at pH communities. So this, of course, community detection is looking for densely connected groups of vertices compared to what you would expect. We use the Louvain algorithm. Something we've talked about is maybe we should use the laude algorithm. The thing that we do, though, is that we flatten our hypergraph into a graph. So we're then any nodes that are in a hyper edge we connect them so that we can have a flattened hypergraph. Now, I'd be really interested in trying some other methods that for community detection, keeping the hypergraph structure. We haven't done that, but that's something that I think we tried, and it was very computationally intensive for one of the methods, which I can't remember now. So if somebody has a fast method, I'm really open to suggestions here for community detection of hypergraphs. But even with this, flattening it, we still are able to get these functional modules that we can interpret. Again, super cool. So if we look at these spatial curves and here is the flower, you can see the loops very clearly. Each of the communities is not picking out, you know, if we had drawn some network, pairwise network, we might not have picked out each of these loops. Maybe we would have, but this is really seems to be picking out. It's not picking out the middle loop separately, right? It's picking out each of these loops as functional modules. If we looked at the spatial analysis alone, it would be hard to detect the structure, but somehow these PH communities identify some structural features that are meaningful. So then the next question is, could we use this on real data? So folded chromatin, these are these long DNA molecules that are known to have lots of noise in the data. This is looking at the pairwise distance matrix of folded chromatin. And the standard way that they look at it is looking at what's very close along the chromatin. So you get these very small, what they call standard domains. With the pH communities, we're finding some structure that encodes some of the local information, right? What's close along the curve, but also some of the structure about what's happening really as a kind of higher order feature. All these features are clustered in some way. And so this is interpretable, and biologists are loving this. They're very excited about it. So excited that, I mean, there's a bit more involved that, and there's other people involved on the paper. So this is the in-prep manuscript on looking at specifically folded chromatin as an application. Okay, so now the mathematician hat's coming on and saying, well, are we really getting anything other than what you can get already with other geometric quantities that I mentioned at the beginning, the torsion, the curvature, or density, which is looking at the point-wise, you look at the fraction of the vertices with distance less than 2, and that gives you some notion of density. So you look at these 800 randomly generated curves with pi nautic, it's generated with pi nautic, and we then compute the curvature, we compute torsion with these 4 different lengths. So curves with 100 vertices, 200, 300, etc. And then we compute correlation between these, the centrality with these other measures, and then the pH community with pairwise distance. And you can see they really don't have that much, they don't correlate that well, which is good. It means we're picking up something else. Okay. So then we looked at the analyzing the pH communities. And what we looked at here is we looked at 4 different measurements. So we have the first measurement is looking at the spatial volume, which is the radius of gyration. And the radius of gyration is also, you can think about it as computing the convex hull. So that gives you a measure of how much space that community is taking up actually in space. Then the next thing we computed was geodesic distance. And this is looking at, for example, if you look at the yellow community, all of the, along this curve, we're looking at what is, here, it's all contiguous, right? You can see that it's all together. Whereas if you look at the red community, we can see that that community is assigned to the same, even though the ends are different. And so that is, has a high geodesic distance. And then the final thing we computed was community size, and that is just simply the number of vertices that are assigned to this community. Okay. So with those, then we wanted to study what we call the geodesic intersection. And this is defined as the p-value of the Mann-Whitney U-test between different sets of vertices in communities. And we have to use this because it avoids artifacts and some sparse, some issues with with just computing the intersection. So we really need some type of value that we, so a low p-value means that there's low geodesic intersection. That would be, for example, the yellow community. Whereas a high p-value means that there's high geodesic intersection. And that would be, for example, a community that, like a red community. Okay, then we take these 4 measurements. So we have spatial volume, geodesic intersection, pH community size, and geodesic intersection. And we then look at it, and we want to look at this notion of a sortativity, which in networks, a partition exhibits a sortativity if similar vertices are more likely to be in the same community, right? And so we've modified this definition and introduced ambient assortativity and intrinsic assortativity. So ambient assortativity is looking at the average ratios between the community size and the volume. Whereas intrinsic, so you can see here that the curves with highly packed communities, so you can see, for example, the second curve and the final curve, they have very highly packed communities. These have high ambient assortativity, whereas intrinsic assortativity is when you have, this is the average ratio between sizes and geodesic size of community. And this is where the curves with communities of small geodesic size have high intrinsic assortativity. So you can see, for example, the second and the third, the 2 middle curves, these both have high intrinsic assortativity, whereas the outer 2 curves, you can see they kind of both have the end. So they have a high, because they have a high geodesic size, leads to a low intrinsic assortativity. Okay. And yeah, so you can kind of think of them as they're localized to dense regions in the curve. Okay. So with this, we then looked at different applications. So we looked at there's this, it's called the Andy Challenge from this paper here in 2020. And this is looking at anomalous diffusion, transport of diffusing particles. And there's these 5 different models: the annealed transient time motion, the continuous random walk, the fractional Brownian motion, the Levy walk, and the scaled Brownian motion. And these are different types of models, and the challenge was, can you develop a method to distinguish them? So we applied the Andy models, so we generated 1,000 of these trajectories, and then we computed these 4 different measures: the volume, the pH community size, the geodesic distance, and I think we called this the geodesic intersection, not overlap. And we computed those, and we could see, for example, that the continuous, the CTRW is very different. And then we actually could use the assortativity methods we talked about to see the difference between them. Then we fed the centrality measures and the pH communities with the weight. The persistence was very important, into a CNN architecture, and we could distinguish 3 of the models. So 2 of them are similar. It turns out that some models are more similar than others, and those 2 were indistinguishable in our using the pH community centrality, pH centrality. But this was a very nice way to be able to distinguish these different models. And then we worked on trying to interpret what these, whether the curves are these very, very densely packed, and trying to interpret that, where you can kind of see that the CTRW, the ambient assortativity here, we can see that those are highly packed, even from just an example of these are very highly packed curves. And then the final application we used was for immune cell tracks. And this is where we looked at neutrophils, which are a type of immune cell, inside zebrafish, and they were cut, so they had a wound, and the neutrophils would migrate towards the wound. And the biological assumption is that the track should follow a persistent random walk. And so when we ran this, there's 3 different types of curves that we studied: the unbiased tracks, the biased tracks, and then the immune cell, these zebrafish tracks. And we could basically not distinguish between the biased and the zebrafish. So it has this a bias based on the wound, and that we were able to pick that up and quantify it with this hyper-TDA with very high confidence. You can see that so that was so we could do that with very high confidence. So I think hopefully I've convinced you that we've been able to look at the 3D structure of different spatial curves, and that hyper-TDA can kind of pull out more information than standard geometric approaches. It also is superior to than only spatial analysis. So we're really looking at the higher order structure, these loops, and using that, as well as all of the techniques, some of the techniques that hypergraph and hyper networks can tell us. It unified some of the spatial and intrinsic distances that kind of, do you study the curve just by what's close on the curve, or do you study it where the points are in space? And we could make sense of both of these notions in some way. It encodes higher order interactions, and in terms of robustness to noise, we know that the chromatin is very noisy, unlike some of the other ones here, where they're generated by some process, which we have a lot of data there. So we're excited to extend this to the chromatin structure. So I think I am a little bit more than 40 minutes. I did start a few minutes late. I wanted to thank our Center for TDA for many fruitful and helpful and stimulating discussions. And in particular, I want to acknowledge my collaborators on this project, and thank you for your attention. Thanks.
Thank you very much.
I'm very happy to answer questions.
So we have some time for questions. As always, people, you can post the questions in the chat, or raise your hand so that I can call on you. And I'm going to get us started with a kind of general question about scaling these things up. So the examples that you were talking about today, those were paths or trajectories that live in 3D space. If my network lived in some sort of feature space that is higher dimensional, like, I assume that this still works, but are there some practical considerations as to like things that might not work as well? Or like, is there something where you'd say, okay, if you have 100 different features, then maybe this is not the way to go? So, okay, so there's no reason why you couldn't study this in some other more general space. We knew a lot about the protein curves, and actually I wanted to apply this to the proteins. Now, one of the issues with the knotted proteins is that those proteins have different lengths. So if you are studying communities that are very different numbers of nodes, or you're looking at centrality, right, and ranking them, and you have different numbers of nodes in your initial data, that's kind of an issue which we haven't resolved yet. And I know Inesi has a student that's looking at that. So she's supervising someone to look at that. So I see no reason why you can't extend to more than 3 dimensions. We don't actually use it's ordered data, but we're not using that except for the minimal generators, which then we don't use. Right. So I really thought we were going to need these minimal generators. I was surprised to see that the Irene generators more or less were fine. So that in some ways is very positive. So it's more general. You can use some other computation. But I think what would be helpful is to have a similar number of points in your data to then analyze. The other thing is that there's no reason why you can't look at other homologies, right? It's just computing one-dimensional homology. There's getting the generators for that. There's
ways to do it. I don't know how to do it for higher dimensional homology. The 50+ page paper that we used that has all the correctness with Irene and for the minimal, that's all, that only holds for one dimensional for the cycles. Again, that's right. I think there's a lot of opportunities, and it would be cool to look at more general data, but those are some caveats, which I guess I didn't, you don't mention all of the limitations in the middle of the talk, I guess. I'm happy to share with all of you.
Yeah, thank you so much. So we have a question from Sohail.
Hello, can you hear me?
Hi. Yeah,
Hi,
yes.
hello. Thanks for such a wonderful talk. I just wanted to ask, can you comment on the application of this thing in image segmentations?
An image segmentation.
Yes. Like if you want to find out like certain structures in, for example, biomedical images. For example, if there's a whole slide image and you want to find out cells, like there are 10 cells, and can you see how this work can be implemented? Because I think there is a kind of remote possibility. I'm not sure.
I mean, I think a lot of there's a number of people have looked at histology and images with TDA. I haven't seen, I mean, we didn't look at that for the hyper TDA, right? So I know that there's certainly been work that has looked at this using TDA for histology. I mean, we've looked at it, but many others have also looked at this problem.
Yes, I mean, as far,
sorry.
Yeah. Sorry. Please go on.
For segmentation,
Yes.
I'm not sure. I mean, it would be very interesting to look at this for segmentation. For example, if you had certain markers that are known to live on the outside of a cell.
Yes. For example, if you have some brain tumor.
Can you elaborate just a little bit more?
For example, you have a like a CT scan and of an of lungs, and there's a nodule, like small little white spot, and I think using this work can we kind of track that white spot in the CT scan because it's a one-dimensional hole or something.
Yeah, possibly. I don't know. I haven't done. I know there is some work on TDA and image segmentation, but I don't know very much. I haven't done that part. Any of the data that we've looked at has already been segmented because that's a very hard task.
In fact, there's not a lot of work towards segmentation using TDA. I mean, there are, I think, 2 or 3 papers that are to the best of my knowledge, because it's kind of hard because of this problem that you addressed in your talk.
Okay. Yeah. Well, if this helps, that would be wonderful. I'd love to hear more. Yeah.
Yeah. Okay. Thanks for the wonderful talk. Thank you.
Thank you for your question.
So perhaps this might be related or not. I was wondering if from doing the hyper TDA analysis, where you showed how you can find these nice protrusions and you can separate them out in your trajectory or in your chromatin line, is there a way that if you had the point cloud but not the trajectory, that you can use any of this to infer the trajectory from it?
Oh, I don't know how. No, I don't know.
Okay.
We don't use any of the trajectory information in the point cloud, right? But we do use it for our analysis.
I thought you're only using it to generate the minimal generators, which you end up not using for.
We don't use it for the minimal generators, but then, for example, when we're doing the assortativity and trying to analyze what's happening with the persistent homology communities. We are then looking at this difference between these two metrics in some way and trying to look at how are things different along the curve versus in space. But we don't have to have that, right? That's like our methods are not critically dependent on that. It was just trying to get a sense of what the communities were picking up exactly, right? I mean, but yeah, I guess I think maybe that would be possible.
That would be really cool. So I like, it seems that there's a lot of cool things to explore this way. So how did you want to add something to that? Because I see your hand is up.
Yeah, I have a kind of, a bit irrelevant question, but since, we have an expert, so I wanted to use this opportunity. So can you please comment on, like, if there are moving structures in a sequence of images, then can you comment on how can we use this TDA to track that thing or something? If I'm not clear.
Can you say the question again?
So if they're like a sequence of images and you want to track some structures in that, so how can we use TDA to do that? Like any hint on that? Like,
so I mean,
for example,
yeah, so what you're asking about time evolving data, right?
Yes.
Because you could have a sequence of images and I think what you're saying is that these are from the same process. So it's a time evolving data set.
Yes.
So there's different approaches that people use. So I think there's a paper by with Chad Topaz and sorry, I'm going to not remember all the names because I didn't prepare for this question, but with like Crocker plots. There's early work by Carlson looking at zigzag persistence and more recent work. And we've put a paper on the archive earlier this year using zigzag persistence to study coral resilience. People have also looked, so Elizabeth Munch has been looking at various different time evolving data sets. Vineyards were initially, so you could track things through, and the picture in the Vineyards paper is actually, it looks like a multi-layer network in some way, but and then there's a paper put on the archive maybe a month ago by Abigail Hickok, she's at UCLA, looking at what she calls persistence diagram bundles. There's a lot of time evolving things, looking at window, right? So these sliding windows with TDA. Yeah, but the thing is that there's no one size fits all. So you have to actually kind of get into the problem and then play and over time get some intuition of what to use for what problem. Sorry, okay, I threw a lot at you, but there's a lot there. So time evolving problem.
Yeah, thanks. That was very helpful.
You're welcome.
And so with that, we're at the hour. So I'll take the opportunity to thank Heather once again for being here and giving us this excellent introduction to TDA and to hyper-TDA. And thank you all for being here. Francisca Echo and I will be in touch at the beginning of next year with more talks to come. And I'm looking forward to see many of you again next year. So enjoy your December and we'll see you soon. Thank you.